Okay, guys, so we just left the stomach and now we're going into the small intestine. So small intestine first, then large intestine. Make sure you understand that the order of the food. Okay, small intestine. So let's look at the small intestine. So first of all, let's talk about what the difference is between the small intestine and the large intestine. So if you just look at total volume, the small intestine is actually bigger. But if you look at the diameter of the tube, the small intestine has a smaller diameter. So that's what we're going to look at. If you look at the diameter of the tube, the smaller one is the small intestine, and the fatter one would be the large intestine. That's the difference. The small intestine has 3 regions. So here would be the stomach here. The very first region coming off of the stomach, that is going to be the duodenum, or sometimes people say duodenum. I think that's more common. I prefer the word duodenum because that's how I pronounce it. Or that's how you would spell it. So duodenum. But again, however you want to say it is fine with me. So there's the duodenum right there. The middle region is the jejunum. And then the last region is the ileum. Now, in a model or a picture, there's no real distinctive boundary between these 3 regions. So the duodenum just sort of becomes the jejunum. Okay, so it's about the first 25 centimeters, but it's not a distinct, there's no specific distinctive structure. But you can definitely identify the duodenum and the ileum based on what they're connected to. Okay, so for example, in lab, if I point over here and it's definitely coming near near the stomach, that's going to be the duodenum. And if I point near here and it's definitely right next to the large intestine, that's going to be the ileum. But for the jejunum, I would have to give you more information. I'd have to say something like, name this middle structure of this organ or something like that, right? So the boundaries are not very particularly distinctive. Okay, so looking at the mucosa of the small intestine, they have what are called villi. Okay, so villi means that they are finger-like extensions that go upward into the lumen. So they're going to stick out into the lumen. Now those villi are going to be on bigger folds that are called plica circularis, sometimes referred to as circular folds. Now, if you're looking at this picture right here, this is a real picture of the small intestine. So if you're looking at these large folds here, they sort of look like the corkscrew, like if you look at a screw, the little ridges on the screw, those would be the plica circularis, the larger folds. And if you look right here, see these finger-like extension that make it kind of look fuzzy, sort of like a carpet? Those would be the villi, okay? Now, coming off of the surface of the cells, so if you go zoom, zoom, zoom a lot, if you look at the surface, the apical surface of the simple columnar epithelial cells, you'll notice that it has these tiny little finger-like extensions here. Those are called microvilli. Okay, so remember that microvilli, they look a lot like cilia, but they are different from cilia because they do not move, they do not create currents, they are not moving food along. Their job is to simply increase surface area. So the functional significance of the plica circularis, the villi, and the microvilli is all to increase surface area. And the reason for that is because the small intestine is where most of the absorption occurs. Absorption means taking nutrients from the lumen, the empty space, and bringing it into the body and putting it into the bloodstream so that you can, give it to the cells. So that's absorption. And absorption is only going to occur at the surface. And so the more surface you have, the more absorption you can get. And this makes it so we're much more efficient at getting nutrients out of our food instead of losing it to waste. Okay, so all of these structures are to increase surface area to allow for a maximum amount of nutrient absorption. Okay, so I did want to mention that, remember that microvilli are micro, very, very small. They're on the little surface of the cells. So structures like these would be villi, not microvilli. Microvilli are microscopic. Okay, so putting in order from smallest to biggest. Oh, largest to smallest. Okay, so the biggest of these would be the plica circularis. Those are the large corkscrew-like structures, the folds. Then you create the villi, which are the finger-like extensions, and then you have the microvilli, which are the tiny extensions on the surface of the cells, also known as the brush border. And the function of all of those is the same, and they're all to increase surface area. So let's take a minute to compare and contrast the duodenum and the ileum. So remember the duodenum, that's the first one that's going to be connected to the stomach, and the ileum is the last one that's going to be connected to the large intestine. Because the duodenum is the very first one, that is where the majority of nutrient absorption occurs, because the food has just been digested by the stomach. The stomach dumps all of that nutrient-rich food into the duodenum, and then the duodenum is going to draw the nutrients out of the lumen and put it into the bloodstream. So you need to be really good at absorbing that food. And so if you look at the villi of the duodenum, they're going to have very long extensions, and they're very elaborate, really complex, wonderful little structures. And that's because of that need to absorb the nutrients. Whereas by the time you get to the ileum, at the very, very end, a lot of those most of that nutrients has already been absorbed, and so you don't need to have quite as long and elaborate villi. But you also need to have Peyer's patches. Now, Peyer's patches are immune structures. They're sort of like lymph nodes, but inside of the intestines. This is where immune cells kind of hang out. So lymphocytes will kind of hang out in these Peyer patches, and they just sort of wait to come in contact with pathogens, and that's how they're going to create immune responses there. So the Peyer's patches are going to be found in the submucosa. So there's the mucosa there. There's the submucosa. This is the muscularis externa right there, and they're going to be found in the ileum and not the duodenum. Okay, so you should be able to kind of recognize the differences between the duodenum and the ileum. Okay, so from the small intestine, we're going to go into the large intestine. So here is the ileum. That's the last part of the small intestine. The valve that controls movement of food from the ileum into the large intestine, that's going to be called the ileocecal valve. And that name comes from the boundary between the ileum and the cecum. Now, the cecum is just this first expanded pouch of the large intestine. So there's your cecum right there. And then coming off of the cecum is a little finger-like projection that's called the appendix, or I think your lab sheet calls it the variform appendix. Okay, so this appendix is right there. Now the appendix is that's also for immune function. It houses immune cells, and what happens is it traps bacteria that might be in this large intestine in the appendix and allows the immune cells time to respond to that bacteria. So appendicitis just means that the bacteria that was trapped in the appendix kind of got out of control, and it's inflamed, and you can actually have a burst appendix. And the problem with a burst appendix is that it is life-threatening, because you're just releasing infectious pathogens all over the entire abdominal cavity, which can be really, really bad. Okay, so yes, if you hurt on the lower left side of your abdomen, you definitely want to get yourself to the hospital. Okay, so again, cecum, first expanded pouch. Coming off of that, there's the appendix, or the variform appendix. Then we have the colon, and the colon has 4 regions, and they're named based on what they do. So you have the ascending colon. Ascending means go up. Then you have the transverse colon, transverse plane, the horizontal plane. Then you have the descending colon, descending means go down. Then you have the sigmoidal colon or the sigmoid colon, and sigmoid means S-shaped, okay? Sigmoidal means S-shaped. See how it creates an S structure? And then the last part of so the colon will then dump into the rectum, which will become the anus, and that's how the waste products are come out. Okay, so let's also look at a couple of the other structures, Hostra and Antennae coli. Okay, so when you look, so here's the intestine right here. Okay, so with the large intestine, the colon of the large intestine has this ribbon of smooth muscle that travels along it. So if you're looking at this picture, see this kind of light structure in the middle? That would be the Antennae coli. And the Antennae coli what it does is it kind of pulls, it's like a drawstring purse, right? If you pull on the string, right, what it does is it makes the rest of the intestine kind of pouch around it. So those pouches that are coming as a result, those are called hostra. So hostra are the pouches, and the Antennae coli, excuse me, is the ribbon of smooth muscle that kind of creates those little pouches there. Okay, so if you're looking at this picture here, these are the hostra. Hostrum is a singular version of that. Hostrum 1, hostra multiple. And then Antennae coli is the ribbon in the very, very middle. Okay, so a lot of people use the intestine, so the large intestine and the colon sort of interchangeably. So what would be the difference between the large intestine and the colon? So remember, the large intestine is a broader term that includes not just the colon, but also the cecum, which is at the very beginning, the rectum, and the anus at the end. Okay, so the large intestine includes mostly colon, but also these other structures, these other regions. Let's look at some histology of the mucosa. So again, we see simple columnar epithelium, just as we saw in the stomach and just as we saw in the small intestine. But instead of creating villi that go up, we have invaginations that go down, just like the pits, but these are going to be smaller and less deep and less wide. So they're instead called crypts. These are called intestinal crypts. Okay, so if you've heard that word before, crypt, it's like Tales from the Crypt. So a crypt is like a grave site, like go down into it. So think of it like going down into the crypts. Okay, and the other thing I'd like you to know is that the simple columnar epithelium, see all of these light colored cells here, those are all goblet cells. Remember that goblet cells secrete mucus. Why would you want a bunch of goblet cells in your large intestine? So if you think about the path of the food, so in the stomach, you got chewed up food going into the stomach. That's called the bolus. And from the stomach, it's going to go into the small intestine. And then the function of the small intestine is to absorb all of the nutrients. Along with the nutrients, you're also going to be absorbing water. So that means that by the time you get to the large intestine, the food is going to be a lot drier. Okay, so you need these extra goblet cells to secrete the mucus to help lubricate and move the food along. So the reason I tell you that story is not to gross you out, but to really help you try to remember that in the large intestine, if you're looking at a picture of the large intestine and you see just tons and tons of goblet cells, that's going to be the large intestine. Okay, so let's just review the different mucosal surface structures that you're going to see in the different organs. In the stomach, you have gastric pits. Those are long, elaborate villi that go, so not villi, I said the wrong word, long, elaborate invaginations, so goes down into it. And then in the intestines, you have villi that go up. And in the large intestine, you have the intestinal crypts, which means they go down into it. Okay, so the way I like to think about this is think about like a sheet cake, right? You got a big old chocolate cake. It's got icing on it and everything. And for the stomach and the large intestine, it's as if you took your finger and you just poked down into that sheet cake all over the place. You made little holes all in that sheet cake. Whereas for the villi of the small intestine, instead what you've done is you put candles all over the top of the cake. Okay, so pits and crypts go down, villi go up. And you can really see this if we had the opportunity to look inside the cat, because if you open up the duodenum of the cat, inside the duodenum, you see like a carpet-like structure. It looks like velvet inside because of all of these villi sticking upwards. So it's a completely different texture. Okay, that's enough for now. I'll do a separate one for the accessory organs.